All right, it's my pleasure today to introduce Dr. Matt Hill, who's a professor at the Hotchkiss Brain Institute at the University of Calgary. Hi, Matt.
Hey, Steve, how's it going?
Great, thanks for doing this talk. So Dr. Hill is really one of the world's foremost experts on the endocannabinoid system. And also recently, he's been very interested in as well in cannabis and THC and other things that are in cannabis that have effects on the brain. He's published numerous papers on these topics in the journal Psychopharmacology. And I think we're just going to have a general conversation about the topic and benefit hopefully from Dr. Hill's expertise. So Matt, would you tell us a little bit about the endocannabinoid system and what it does in the brain, and then also how THC kind of interacts with that?
Okay. So the endocannabinoid system is kind of like the brain and body's endogenous system that has similarities to THC from cannabis. And I think the easiest way for people who aren't familiar with this system to understand it is always liken it to endorphins, because everyone seems to know what endorphins are. And so endorphins are like the body's own version of morphine, essentially, and it acts on opiate receptors in a similar manner that like morphine and heroin and other opiates do. And so the THC is the psychoactive constituent of cannabis, and it acts on the endocannabinoid system, which is a collection of receptors and molecules and enzymes that the brain and body possess. And the endocannabinoid molecules themselves are biologically derived lipid molecules that come from membranes that act on the same receptor that THC does. And essentially, the way that the system works is cannabinoid receptors are kind of weird in the brain in the sense of their localization and the way they act, because it's kind of opposite of most other neurochemical systems. So we always think of neurotransmission as tending to be in kind of an anterograde direction. So all the prototypical examples we always talk about is a neuron releases a neurotransmitter into the synapse. It crosses the synapse and acts on receptors on the postsynaptic neuron. Whereas in the brain, the cannabinoid receptors are almost all exclusively localized to the presynaptic terminal. And when they become activated by either THC from cannabis or endocannabinoids, the main function of activation of this receptor, at least the main consequence, is that it terminates or inhibits neurotransmitter release. And so in the context of endocannabinoids, how this tends to work is that a neuron will often release something like glutamate or something that has an excitatory component to it. It will trigger activity of a postsynaptic neuron. And in response to that activation, the postsynaptic neuron will make these endocannabinoid molecules very rapidly, usually through calcium-dependent processes, but not always. And it will release these endocannabinoids kind of into the synapse, where they act as retrograde signals. So they actually go backwards. So they move from the postsynaptic cell to the presynaptic terminal, where they activate the cannabinoid receptors. And when they activate them, this turns off ongoing neurotransmitter release. So in many ways, it's almost like a buffer system or a thermostat. So if a postsynaptic neuron becomes too excited, it makes lots of endocannabinoids. They then turn off the afferent input to gate the excitability of that cell, and kind of the prototypical sense of how an endocannabinoid system would work.
So it's kind of like the brakes on the system in some ways. Is that fair to say?
Yeah, it's I mean, like, so some systems like dopamine has autoreceptors where it self-regulates itself. So it has some presynaptic receptors that sense how much dopamine's in the synapse. If there's too much there, it turns it off. Endocannabinoids kind of do this in a broader sense for like large scale levels of activity in the brain, but they can also inhibit they can also regulate inhibitory neurotransmission. So in that situation, they actually act as a means to increase gain on a synapse or increase the strengthening of a synapse by removing some local inhibitory tone. So they have very complex actions that are super synapse dependent. And there's very different scenarios that will trigger endocannabinoid release to modify synaptic strengthening or weakening in different capacities. And how THC is a little different is obviously if someone consumes cannabis and therefore THC gets into their blood and into their brain, you lose that refined discrimination of where endocannabinoids are kind of having patterned release to explicitly impact specific synapses. When someone consumes cannabis, THC just kind of floods the brain and therefore will saturate and activate whatever cannabinoid receptors are available for it to bind to. And so the effects tend to be much more indiscriminate than what you get from very intentional endocannabinoid release at synaptic subcompartments.
Interesting. So, you know, one of the things that you mentioned there was that THC is the psychoactive drug in cannabis. And obviously it's having these modulatory effects, probably a bit different than kind of the endocannabinoid system in the ways you just described. But is that the only psychoactive drug in THC? I mean, you know, I've heard that there's other drugs in there. So CBD, for example, cannabidiol, people talk a lot about, and you can buy all over the place for everything under the sun. Basically, it's supposed to be good for. Also, people talk about terpenes and things. So like, what is it? Is it really just THC, or are these other drugs and compounds actually like modulating that or having effects on their own?
I mean, I think what we would take from pop culture is we'd certainly assume that there's got to be some modulatory effects because, people can consume 5 different strains or types of cannabis that all have pretty comparable levels of THC in them and yet report like wildly different psychoactive responses to them. And so some people say this one makes me more activated, this one makes me more tired, this one makes me more hungry. So people can have really varied responses to different types of cannabis that have similar levels of THC, but obviously vary in other chemical compositions. So yeah, obviously CBD is the one that gets the most attention and discussion. And yeah, it's the cure-all for everything under the sun at this point, it seems. CBD is an interesting one, and it's probably outside of THC had the most attention to it in terms of what's been isolated and studied from cannabis. It doesn't act like THC. So THC is like a specific direct agonist at cannabinoid receptors. So when it binds to it, will just activate a cannabinoid receptor. CBD does not do this. So, and that is probably why CBD doesn't produce a kind of intoxicating effect the way that THC does, because it's not activating cannabinoid receptors directly. It may interact with the endocannabinoid system still. So there is certainly evidence from different areas that suggests that CBD might potentiate endocannabinoid signaling kind of indirectly, either through blocking its metabolism or enhancing its production, or just making it hang around in the synapse for longer. So that CBD could be doing that. But again, because it's endocannabinoids and not THC, you still don't really get this intoxicating effect because you kind of need that blanket activation of CB1 receptors, probably throughout the cortex, I would guess, to produce most of what we think of as the kind of prototypical cannabis high. But at the same time, CBD also seems to actually interfere with normative cannabinoid receptor signaling. There's some evidence to suggest that it may bind to a different site that's not the main binding site on the receptor, something we call allosteric binding site. And when it binds to that secondary binding site on the receptor, it might actually compromise the ability of THC to activate CB1 receptors. And this kind of goes with the general notion in the public that CBD has this ability to counter the effects of THC. A lot of people seem to think that if they have CBD, it might reduce some of the aversive effects of like high-dose THC. There's some evidence in humans to support that. I'd say it's a little dicey for the most part. I think more often than not, what it is because at a biosynthetic level in cannabis, THC and CBD both use the same precursor. As soon as you breed a plant to be high in THC, you inadvertently make it a low CBD plant because the precursor has to shunt through one of the 2 enzyme pathways. And so any plant that's a high THC plant by nature is going to be a low CBD plant. And so the converse of that is obviously if you have a plant that has reasonable levels of CBD in it, by nature, it's going to have lower levels of THC. And so I think more often than not, people tend to consume CBD enriched weeds that have less THC in them. That's probably why they feel less intoxicated by it, because the really controlled studies haven't been the most convincing to suggest that CBD has this counteractive effect on THC. But it can kind of do some other things. It might interact with adenosine signaling, it seems, where it will enhance its signaling. And adenosine, we know, is important for kind of things like sleep. And that could maybe explain why some people benefit from sleep with CBD and stuff. As for the other thing in cannabis, I mean, there's like dozens and dozens of other cannabinoids that we basically know nothing about. We don't really understand their pharmacology in any real way. None of them seem to be strong cannabinoid receptor agonists like THC at all. So THC really seems to drive the majority of the kind of classic high we assume from cannabis. But they probably are doing something to modify it. I mean, through indirect means, whether they're influencing metabolism of THC or they're influencing other neurotransmitter systems, like they may indirectly impact GABA or glutamate signaling in a way that could interact with the cannabinoid receptor activation effects of THC to produce a slightly different high.
And this is the idea of the entourage effect, right? This is
Yeah,
from like
exactly.
the Shulams kind of stuff. Like the terpenes or something, for example, they don't do much by themselves, but when THC is there, they kind of modulate that and change it in some way. Is that basically right?
Yeah, and there's been a bit of work that's come out of Ryan Vandy's lab, especially at Johns Hopkins, that's kind of looked at like isolated compounds like limonene or beta caryophyllene or things like this and see how they influence kind of subjective effects to THC. And some of the data he's got actually, I would say, looks like it is starting to suggest that there could be interactive effects between some of these terpenes, and they may indeed influence cannabis high. And I mean, I think from a kind of recreational user perspective, it's interesting because the terpenes are what dictate the odor often. So, I mean, some cannabises have kind of a citrusy smell. It tends to be limonene, or some have like a pine tree smell. And there's another terpene called pinene that drives that. And so you could assume that if these terpenes do actually produce interactive effects with THC that influence the high, that someone could associate the smell of that kind of cannabis with this type of high. And so people start becoming very selective on the strains and flavors of cannabis they like, because they will start to associate certain odors with certain outcomes of the intoxication that they prefer more or less than others.
And then there's also like expectancy effects that come from that, right? Because if you smell your cannabis smells like this, and you've smoked that kind of cannabis before and you had X effect, maybe you're going to have that effect again, just because, not because like the actual compounds do anything, but because you sort of expect to feel a certain way, and THC then may kind of allow you to feel that, you know, sort of like your high may sort of be pushed in that direction by that expectancy.
Yeah, cannabis is real. I think out of most of the drugs that are used recreationally, I think cannabis is certainly the most susceptible to expectancy bias in that regard, because it has this rich mythology in pop culture about what people think about different strains and this and that, and certain smells dictate this. I mean, I think even more than cannabis and THC, though, the expectancy effects for CBD are massive. And they've just reached this kind of critical point now where so many people have heard of people taking CBD and having benefit in some capacity that I mean, it's hard to believe that that's not accounting for a large majority of what the effects that people see of CBD in the general public. There was a study that just came out very recently that actually finally tested this really well in a double-blind crossover manner, where they either gave people CBD or they gave them a placebo pill that they told them was CBD. And so they all assumed they were getting the same dose of CBD. And whatever effects were seen with the CBD treatment were seen exactly the same when they were given a placebo that was told to be CBD, as opposed to if they knew they weren't getting anything. And so it's very clear that expectancy bias for a lot of the cannabinoid effects is probably accounting for a huge majority of it. And it's really hard, I think, on an individual level. If someone knows like 4 or 5 people whose opinions they trust, who tell them, I took this and this did this to me, they're just going to believe it. And it's very hard to overcome that expectancy of what you think is going to happen in that regard.
Yeah, this is a very powerful, I mean, this is maybe also known as a placebo effect, essentially as an overlapping term, at least. You know, it's not like, you know, a lot of times I think people sort of think placebo effect, that means nothing happened, but that's not true. Like, the brain is a very powerful thing. And if you expect something, to a drug or some compound you're taking, to have a beneficial effect, like, it might have a beneficial effect. It might actually help you, but not because of the drug, but because sort of the way your brain works, like in terms of like expectancies and stuff. And then also just throw out there that, like, when it comes to CBD, you know, you're probably talking about really well-controlled scientific experiments where the CBD is sort of molecularly isolated or synthesized independently from THC. But most of the stuff that's out there is going to be extracted from a plant, which has some THC in it, not very much, but a little bit. And maybe a little bit goes a longer way if, for example, your liver is metabolizing CBD the whole time, and that little bit of THC can kind of hang out longer and essentially have THC-like effects, which we definitely know is a psychoactive drug. Does that make sense, or am I making?
Yeah, no. People underestimate this. I mean, I think a lot of people don't realize like how low the doses of THC, especially when taken orally, which is how most people take CBD, for example, like in an oil or something that they consume orally, not through a vapor or a pulmonary route. You don't need to take much THC in an oral capacity for it to have a psychoactive effect on you. So, I mean, some people will feel as low as 2 to 3 milligrams of THC. 5 milligrams for the majority of people, they would detect that. And 10 milligrams, you have people who are starting to have aversive responses to. Now, a lot of things try and claim like, oh, there's super low CBD dosing because it's something like 20 to 1 CBD to THC. And that sounds like you're really talking about a very low amount of THC. But from a clinical standpoint, despite the kind of, you know, people in the general public probably take CBD dosing at incremental levels that are really not doing anything because it has such poor bioavailability. But in a clinical setting, these doses are usually 300, 600, 800, over 1,000 milligrams of CBD that people are taking to actually have clinical benefit. Some of the epilepsy studies go over 2,000 milligrams. So in that context, you're talking about something that's 20 to 1, you know, at 1,000, you're still ending up like, there's a reasonable amount of THC that is actually getting through in there. You're well into the dose range where people are going to have psychoactive effects. Like even if you were taking 100 milligrams of CBD, you're still getting 5 mgs of THC. That's not trivial.
Yeah.
And so it is hard to disentangle that because so few things have actually been done with kind of molecularly isolated CBD in that sense.
Really interesting. And so like one of the things that you mentioned also was that, people think, this strain does this. This is sort of my sativa that keeps me really clear-headed, or this is my indica that makes me drowsy and helps my pain and helps me go to sleep and stuff like that. And it may well be that there's compounds that differ between strains, let's say, that have effects like entourage effects or whatever else. But one of the other things is that I think we know that THC itself can have like really variable effects. So even in the absence of these other compounds, for one person, it might make them have a panic attack. And another person, it might make them feel much less anxious. So what is going on with that? Like, how can you explain that?
Yeah, THC, it is wild again. I mean, you might get a little bit of this with alcohol. Like you do get people who have differential responses. Some become aggressive, some become chatty, some become sad. Like people have like a little bit of a psychological difference. But with cannabis, it's really fascinating to me that you can have a drug that like is this one molecule that we know how it acts, and yet it has such diverse impacts on people. So I mean, here in Canada, because cannabis is legal, there's obviously ads about safe use the same way there is about alcohol that you would see in the States as well. And so it's interesting, is one of the more recent ones they've done is they have a group of like 5 people sitting around a table, and they show one person's getting like kind of wigged out trying to figure out what's going on, and one of them's like falling asleep, one of them's laughing, one of them's eating, and they're just making a point about how different cannabis can affect people. And so to be careful when you're sampling it or trying it, just so you know that you can't expect, if you haven't had experience with it, you may not know how to expect how it's going to affect you. And this is one of the things that we've always found fascinating in the field. I mean, there are some base things that we can kind of understand that do influence this. And so one of them is definitely dose. And so there is clearly a biphasic effect with cannabinoids. And this is well established now because we've seen it in all the rodent and animal studies, as well as the human studies, that we do know that like lower doses of cannabis, lower doses of THC kind of produce this plethora of effects that most people would consider to be more on the positive side. So people will feel relaxed and less anxious and less stressed, and they'll want to eat tasty food and they'll laugh more. And for the most part, people view it as a positive experience. And people who have that kind of response to cannabis do tend to continue using it because it's a positive experience for them. But we know that as the dose can increase, we get this flip where it's not just like a muted version of what the lower dose effects are. It's like you were saying, it's the polar opposite. And so you can go from, cannabis typically for people at low doses is anti-anxiety. It goes to high doses and you get pro-anxiety, and you get panic attacks, and people get really paranoid sometimes. And we know that we've seen this flip. We see it, like I said, we see it in the rodents as well. But because we see it in the rodents, that's actually given us this ability to disentangle the neurobiology of it a little bit more and understand it. And one of the things that we've learned from this is because, as I was saying at the beginning about the endocannabinoid system, there's no such thing as a cannabinoid neuron. The way that there are dopamine neurons or glutamate neurons, cannabinoid receptors regulate everything else in the brain, basically, because the receptors are just kind of promiscuously spread over all these other axon terminals. And what we know is that cannabinoid receptors on glutamate terminals, when they become activated, they'll reduce glutamate release, and glutamate is excitatory. So if we look in a brain region like, say, the amygdala or something like that that's involved in regulating behavioral states like anxiety, we know that these parts of the amygdala becomes hyperactive in individuals when they're anxious, when they become stressed out, if they have post-traumatic stress disorder or an anxiety disorder. And we know that as anxiety subsides, activity in the amygdala tends to decline. And what we know is that cannabinoid receptor activation on glutamate terminals in the amygdala will reduce the release of glutamate and therefore reduce the excitability of a lot of the neurons in the amygdala. And that does seem to account for the majority of the anti-anxiety effects that are produced by cannabinoids. But when we look over at the next set of neurons around there, you've got clusters of GABAergic interneurons, which are providing tonic inhibition on these same cells. And so if you get cannabinoid receptor activation on these GABA terminals, we're reducing the amount of GABA that's released. And so that means there's less inhibition on these neurons, and now they're actually becoming more active. And so that actually seems to contribute to the generation of anxiety and the induction of things like panic and paranoia that would probably come in a human setting, at least, when they flip into the aversive side. And so that dose threshold seems to be able to predict the difference between having kind of a positive effect versus it trickling into a negative effect. And I think if we kind of think of that, one of the things that we can understand about the context of nowadays cannabis, let's say, versus oldie time cannabis from the 60s or 70s, when it was like, back then cannabis was like 2 to 5% THC. Whereas nowadays it's like 25 to 32% THC for the majority of the stuff that you'd find in a commercial venue. So, you go back and watch like a Cheech and Chong movie from the 70s, and they've got these gigantic joints because at 2 to 5% THC, someone could manage dragging on that thing and still not overwhelming their body with THC. And if you were someone that maybe had, different cannabinoid receptor expression, like you were on the upper end of the spectrum with more receptors, you had a lot of given there to be able to titrate a dose before you maybe cross that line of this is a place that's positive and not a horrible experience to, oh my God, the world's trying to eat me kind of thing, where it flips into this other experience. And I think what we see nowadays, because cannabis has become so much more potent than it used to be, that threshold can be very trivial because the amount of THC that gets into your system from a single bout of consumption, like if someone's smoking a joint or a vape pen, one puff is going to hit one level of THC, and that might be good for them. And another puff in and of itself might actually just be too much. And so that threshold from when someone goes from this is a good experience to this is a horrible experience can really come down to being 1 or 2 puffs with modern cannabis because of the potency, especially if they're not very experienced in it. And so I think a lot of the reasons why a lot of people nowadays tend to have more aversive experiences or variance in these experiences to some degree could be accounted for just differences in their own cannabinoid system biology. Some people are going to have more receptors, some are going to have less. One of the things that's interesting is it does tend to be a tendency for women to have a greater likelihood of aversive responses to cannabis than men. And there is some suggestion that women may have more CB1 receptors on GABA. So that may explain that they have a narrower threshold. That being said, they also, work you guys have done as well as what we've done looking at the metabolism. There's a very well-established sex difference. Females tend to make more of a potent metabolite of THC. So not only are they making more of a metabolite that's more potent, maybe they have more receptors on GABA terminals, and those two factors together just might make the window of transition of a low dose anxiolytic, relatively positive effect to a high dose anxiogenic, aversive effect happen quite easily.
Interesting. And I wonder if another aspect of this, so the dose thing is very well taken. I think that's absolutely right. But I wonder if there's another thing going on here too, which is sort of like given that cannabinoids in the brain normally and THC hacking into that system basically is a fundamentally modulatory. It doesn't do much by itself. It just shuts down stuff that's already going on, like shuts down release of neurotransmitters that's occurring at the time, especially with THC. Whatever's happening, it's going to shut down release if there's cannabinoid receptors there. I wonder if sort of what's happening in the first place is part of this puzzle as well. So let's imagine that you are a person that's very anxious in the first place. Maybe you have a lot of glutamate release in the amygdala, and then you smoke a joint and you have the THC inhibits that glutamate release, it reduces your anxiety. Whereas if you're sort of sitting there and you're not anxious, your amygdala glutamate is really low, maybe your GABA is going in kind of a protective sort of way, and now you smoke cannabis and perhaps then that disinhibits these neurons and now you're really anxious. I wonder if that might be part of this as well. What do you think?
Yeah, no, I definitely think that the state dependency of that, I think, is a big thing. And I think it is interesting because for a lot of people that do have high anxiety, they do tend to more generally find cannabis to be anxiety reducing. And that is social anxiety disorder is one of the biggest predictors of someone transitioning from recreational use to a cannabis use disorder because of the fact that they tend to report that it really helps bring that down and makes them feel a bit more comfortable and a little less anxious. Whereas, yeah, a lot of the people that tend to have kind of panicky reactions, it does tend to be in relatively ambient conditions where there's no reason why they should necessarily be stressed. That being said, there does also seem to be an order effect as well, which I think reflects some kind of state dependency. So if someone has been stressed out, people will often use cannabis in the aftermath of stress to reduce, feelings of anxiety and feelings of stress. And that seems to help bring them back down. But if someone has cannabis before they get stressed, sometimes it's been shown that it actually can potentiate that quite a bit. And that again may just have to do with the state of how their brain is processing the information that's coming in. You're going to get dumps of things like noradrenaline and dopamine with highly salient and arousing events. And that may interact with how cannabinoids are working in a totally different way. There was this wild study from the 70s, which they would only do this stuff in the 70s, where they gave people THC before putting them into dental surgery and then measured their anxiety. Everyone had a panic attack because they got super high and freaked out when they went into dental surgery. And you're kind of I can't even see something like that happening nowadays. But I found it fascinating because it's, again, one of these things where had they just been there I mean, you kind of wonder, had they given the THC after the dental surgery, would the people have had a similar reaction, or would they've actually had the opposite, where they felt more relaxed by it? And was it just a priming effect? Because, maybe, they were in ambient conditions and they take the THC, so it's reducing this GABA inhibitory tone. So it's maybe priming them to suddenly react to a stressor or something being added on, because now they don't have that strong inhibitory tone, kind of keeping a lot of things quiet in the brain. But again, these are tricky questions to ask, like, especially with the complexity of it. It's just because we have relatively well-established behavioral phenomena, we can make guesses as to what's going on in the brain, knowing the biology of how the cannabinoid system works.
Very interesting. Yeah, there's just a huge amount of complexity here, and there's obviously just a ton more we need to know about this, especially given that, these things are, cannabis is being legalized everywhere. It's legal in Canada now. It's legal in my state, California, as well as I think about half the other states in the US. And I think this is coming for the rest of the world as well. So, kind of understanding how these systems work and how they interact with the endogenous systems, both for purposes of developing treatments for people where these drugs might be useful, as well as just for understanding what happens when people use them more. We really need to kind of know more about that. And with regard to that kind of topic, one of the things that people talk about a lot is that whatever happens in your brain when you're an adult and you are over 18 or 21, whatever the rule is, and you are smoking cannabis voluntarily, that has effects, and it can be an addictive drug, et cetera. There's like ups and downsides of these kinds of things. But one of the things people do talk about is that when you use these drugs younger, there's a chance of, given the fact that your brain is still developing, it could be that some of those developmental processes are cannabinoid dependent, like involving the endocannabinoid system. And by flooding the system with an external drug, those things could be disrupted in a way that wouldn't just have effects while the drug's on board, so like for a few hours or days, but in fact for forever, because you're sort of in this developmental window where, like, whatever happens then, like, it is what it is, and it's not going to kind of like, you're not going to have another opportunity to wire up your brain the right way or something after adolescence, for example, is over, or prenatal development's over. So I wonder if you could comment a little bit about developmental aspects of cannabis use. Like, do we have good reason? Should we really be very worried about this in humans? Where's this evidence coming from based on the animal studies and the human studies? Like, what's your take on that topic?
Yeah, it's very mixed. So I think if you start with the fundamental biology, I can certainly say, yes, we know the endocannabinoid system does have like a really important role in brain development, certainly in utero. Its exact role during adolescence, I would say, is a little less clear at this point. We haven't really mapped it out, but there is a large body of really, really elegant work for the kind of early organization of synaptic formation, especially cortical. So we know it's really important for like those early kind of corticothalamic, cortical connections. And so cannabinoids act as a guidance cue. And so what we know from, like, especially Tibor Harkany and Yasmin Hurd and a bunch of others have done work like this, is like axons will release endocannabinoids as kind of a signal that tells it where to go to form appropriate synapses. And so obviously you can think that we know from the work they've done that if you take the cannabinoid receptor offline, it really messes up where all these synaptic connections actually mature into. And so that's going to affect the development and the organization of a brain that will go on permanently. And there is work that they have done, especially Yasmin, in terms of especially looking at some of the human tissue as well to see this, that they have seen there is some disorganization with the way that some parts of the brain may organize. Certainly, she's got some work on GABA neurons in the hippocampus that's interesting. And a lot of the animal work that they've done has also shown that you might get like misappropriate synaptic connections forming in the presence of something like THC. The human side, in terms of kind of the long-term studies of prenatal exposure, have been obviously harder to do for various reasons, because as is the case for many things, I think there's a lot more variables that tend to track. Like if you've got someone who's openly using cannabis while they're pregnant, I mean, there is a proportion of women who use cannabis unknowing of their pregnancy for the first few weeks, because women often don't realize the pregnancy until they're several weeks in. And so you have some incidental exposure that's happened in that window that could be having an impact. And the proportion of women who then continue using cannabis for the whole pregnancy is a lot less. You'll have some that may carry on for things like nausea and whatnot. But that may represent a different subset of people, that there are other socioeconomic, nutritional, life stress variables that could also interact with the prenatal cannabis use. And so the first study that really did this was this huge cohort out of Ottawa up here in Canada. And they found some effects as the kids grew up, who had been exposed to cannabis in utero. They weren't super, super robust. There was some stuff with attention and some cognitive functioning that came out during development. They did find that the kids tended to be more likely themselves to smoke pot, but again, it's very hard to disentangle that because these kids would have obviously grown up in like highly cannabis-friendly environments.
Yeah, mom was smoking weed during pregnancy, so she's probably not super against it, and they warning you heavily against it.
And the description of a lot of the women that were in that study from people I know who've been involved in the study was they were kind of like the barefoot earth mothers. Like it was a very different phenotype of women than a lot of the control group women were. And so the comparisons can become a little difficult to isolate down and say, oh yeah, this is explicitly due to the cannabis exposure. And that's obviously where the benefit of the animal models come in, and you can start to see some stuff. And we've started to see some of this come out more recently. There's definitely been some work from Miriam Mellis's group that really nicely has shown some impacts on dopamine neurons and sensitivity of dopamine neurons into development and in adolescence, and some changes that might be hardwired. And they tend to be more male specific. And I've really found the work that she and Joe Cheer and others have been doing in this area to be really interesting, because if we look at some of the large scale human data that's now been collected in Canada, there was a paper that came out not too long ago that did find increased risk for autism in the offspring of mothers that had been using cannabis. And in the ABCD cohort, they also found an increase in the proneness to psychosis in the offspring of mothers that had used cannabis. Now, what I find interesting about those is that the proneness to psychosis is obviously potentially linked to changes in dopamine neuron function, but the autism aspect of it as well is also male biased. And since the other domain where a lot of the animal work has shown reliable changes from prenatal cannabis has been under conditions of social behavior, again, specifically in males, I think there's kind of like an accumulation of evidence from a lot of different like epidemiological, basic science, some controlled studies that have started to suggest that, yeah, okay, there might be some greater risks for neurodevelopment, some greater risk of neurodevelopmental conditions like autism spectrum disorders or schizophrenia. There may be more of a male bias in terms of the vulnerability here. So I think we're at that point now where there's, you know, as good scientists, we should be good observationalists. So we kind of look at this and say, okay, there's something here that now is worth pursuing. And I think that in the next few years, it'll be really interesting to start seeing if we can get more refined questions and start looking at those to see if the prenatal side of it, if there is something there that's genuinely concerning that we need to be able to create appropriate warnings for. The adolescent stuff gets really difficult because almost all the human studies, again, we're looking at very different representations. Like, you know, a lot of the studies are looking at kids who start smoking cannabis around 12 or 13. There's typically a lot of other things going on in their lives that are not just using cannabis. And so individual studies have found, you know, effects on brain development or relationship to various onset of different mental illnesses. However, certainly in the context of brain growth or neuropsychological functioning and just, you know, people who use cannabis as teenagers and then looking at them as adults, the large scale meta-analyses or really large population size samples have really struggled to actually find consistent effects. And so my view on it is that I think realistically, there's not really been well controlled for patterns of use, frequency of use, potency of product being used. So I think there's a lot of variables we can't say here. And so I think that there probably is a risk threshold in terms of cannabis use during a period in adolescence, in terms of age of onset or frequency of use or things like this that could have more defined and reliable adverse outcomes related to it. I think with the way that a lot of teenagers use cannabis, which is more infrequently, you know, a couple times a month, maybe on a weekend at a party or something, and not regularly, I would be hard pressed to believe that that would probably translate to a lot of long-term kind of neurodevelopmental impacts, just because, I mean, we have an endocannabinoid system already. We're not, it's one thing to kind of permanently obscure that system and block it by like using cannabis as a wake and bake to when you go to bed and you're smoking all day and all night. So the pharmacological drive that's hitting that system is fundamentally different. But I would imagine that like really kind of brief intermittent use that's occurring in a more sporadic recreational manner as teenagers would use it is probably not having as much of a huge impact on the system. And it'd be really hard to understand how just having a broad agonist kind of hit everything at once versus the way that pattern endocannabinoid release would go on like intermittent basis would have this kind of permanent effect. So I think there is risk there. I think the thing is we need to really dial down is understanding the risk, and does it interact with other things in our life, like nutritional status, obesity, life stress, other things like concussion from sports injuries. Like, are there other things that are going on in tandem that could amplify the risks associated with cannabis during adolescence? And I think that because it is just something that's so, like in Canada, it's like 50 to 60% of the teenagers. I assume the States is pretty close in terms of the frequency of use, and legalization, at least in our hands, hasn't changed that dramatically in any way. Teens tend to use before legalization the same way they do afterwards. And I think for most of that I've seen in the States, that's generally true as well. So I don't think any of this is necessarily related or consequential to legalization, but in terms of the cannabis use, I think we really need to understand where those defined risk windows become more and more relevant to be able to give kind of appropriate public health messaging, as opposed to, you can take the abstinence approach, but we learned from the war on drugs and all the stuff from when
Doesn't work.
That did shit all. Like, that didn't stop anyone. If anything, it actually increased drug use. So the worst thing to do is tell a teenager to not do something, because the obvious also that is they're going to go do it. So I think you kind of got to inform a teenager realistically and not talk down to them in a threatening way to be able to create appropriate messaging for them. But I mean, I think we need to understand things really in the complexity of them before we can start doing those tailored messages in a real way.
Absolutely. And I think demonizing these drugs that are obviously not like killing everyone left and right, there is no reefer madness, et cetera, like we sort of like heard about from at least the US government in the in the 40s or whatever it was. That's when you're hearing one message that this is a terrible thing, and you see other people doing it and they're not terribly affected, like this is can sow just distrust, which can have lots of different negative consequences, obviously. But that said, we have a whole lot more to learn about this kind of stuff here. We don't know what the threshold is. We don't know if it's just that people who smoke a ton of weed when they're really young end up out with negative outcomes because of doing that, or because they already had stuff kind of going on in their lives that caused them to smoke a ton of weed at such an early age. Yeah. These are really tough questions, and I think we need to kind of push forward on all fronts, both with the human studies that can try to dissociate these variables, as well as with our animal studies, which, despite their limitations, can at least establish causation of these kinds of things as well. And so we have lots more work to do, and I'm hoping that people like you, including you, publish some of that work in psychopharmacology as well. Journal's very interested in this kind of stuff, and I think there's plenty more to come. And honestly, this I feel like is probably going to have to be part one of this interview, because I have so many more questions to ask you and things that I'd like to talk to you about, but we're out of time. And with that, I'll then thank you for your time, and I really appreciate you having this conversation with me today.
Yeah, no, it was a blast. I'm happy to come back for part two. I think my blind dog is like watching us in the background. She's like leaning off the couch, peering over now. She just hears the voice and she's like, "What's going on?"
It wasn't that she smoked weed too young, was it?
No. She just had no eyes when she was born, so she had her own set of problems.
Great. Okay. Well, thank you again, Dr. Hill. This was really a great talk, and I look forward to a part two someday.
Yeah, sounds good. Thanks for having me.